Welcome everybody to today's Matter to Life lecture series, a lecture series where we present you the different research of the Matter to Life Fellows faculty. And they are distributed all over Germany, as you can see here on the map. We currently have 14 locations where our fellows are located. And in October, I just told Andreas, there might be some new fellows joining the Max Planck School faculty, so we might even have some new locations. So keep posted and you will see who's coming and joining the faculty. And today it's my pleasure to have Andreas Janshoff here. He's at the University of Göttingen. And as always, we have two questions for you, Andreas. So first of all, I'm very happy to have you here. And we have two questions to get to know you a little better before we dive into your research. And with this, I'll start with the first question. And this is, can you tell us about your current role at the Max Planck School Matter to Life and how your work contributes to the broader Matter to Life field?
Yeah, thank you very much, Jan, and also for the possibility here to present some new data from our group. Yeah, my role is basically divided into two things I'm doing here in this Matter to Life school. One is, of course, teaching. Teaching means that I'm teaching biophysical chemistry classes, but also master classes for the Matter to Life students concerning spectroscopy with a focus on biological molecules. And research-wise, we are basically at the interface of bottom-up and top-down approaches, while we are trying to merge these two approaches into a more holistic view of biophysical processes happening in the cell.
Thank you very much. And then the second question, so what are some of the most significant challenges you have encountered in your career so far, and how did you address them?
Yeah, I mean, here as well, one has to be more specific as far as the challenges are concerned. There are, of course, typical research challenges as they occur all the time in lab or while trying to understand a specific problem, but also, of course, administrative hurdles that have to be overcome. And this is basically this tug of war between administrative duties and the freedom of research, and I guess that is maybe the challenge I'm facing right now for the last 10-20 years, in fact.
Mm-hmm. Right, and thank you very much for this introduction and getting to know you a little better. And now we're very curious to hear about your current ongoing research, and looking forward for your talk titled The Role of Polarity in Tissue Mechanics. And with this, Andreas, stage is yours.
Yeah, thanks a lot, and a warm welcome from my side to this talk. And some of you might have heard part of it already in a different context, but I want to let's say highlight some of our more recent findings, and that's the reason we are addressing now cellular polarity of epithelial cells specifically. And as you all know, cellular mechanics is pivotal in many biological processes that comprise, of course, the cell functionality, the fate of cells, for instance, how stem cells develop in a specific environment. They are recognized, for instance, the stiffness of their substrate and differentiate accordingly. That adhesion and growth needs to be mentioned. It's, of course, depending on the substrate, and also, of course, the cellular mechanics, the response of cells to external stimuli also depends on that state. Of course, cellular mechanics is important for motility, cell division, and also on larger scales for collective phenomena like the epithelial to mesenchymal transition, wound healing, tissue formation, and cancer. And since we want to span a little bit the length scale from single cells also to tissue, we will always come back to these different phenomena. When you want to study the mechanical properties of cells, one thing that comes to your mind immediately is, of course, you have to deform the cell in order to get a response. And from that response, you infer the elastic or viscoelastic or poroelastic properties. And one of the early data has been obtained in 1964, appeared in this Journal of Experimental Biology, and the very last one is from Mitsuko Yoneda, tension at the surface of sea urchin egg, a critical examination of Cole's experiment. And this is an experiment which we actually also use here with much smaller cells. And this is shown here. We actually try to glue cells to an atomic force microscopy cantilever and then deform it by squeezing it in between a substrate and this cantilever and obtaining a force response. And that allows us to understand how cells react when being deformed quite substantially. Here in this case, of course, just a cell in suspension, so not adhered to a surface except that it's glued to the cantilever. So the typical force response we get here is divided into two parts. So we use the force ramp to compress the cell in between these two plates, and then we get, of course, an increase in force, and then we stop at a certain height. So that's a height clamp experiment, and then measure the force relaxation, and that's this yellow part here, the force relaxation. And in this relaxation part, you can immediately see that there's some kind of viscoelastic, some memory effect here. If you had just an elastic entity, you would get, of course, no relaxation at this point. So we divide this curve into these parts and try to describe them. And one way to describe it is to write down a Hamiltonian that allows us to get a model for that cell, and this is nothing else than a shell filled with a liquid. And the Hamiltonian is divided into three parts. As you can see in the bottom, there is, of course, the bending part, that is the bending penalty, if you wish. And then there is a tangent part, and this has to do with stretching of the entity. So whenever you deform a spherical cell, you will get a stretched shell in response. And then there's, of course, the last part, which is the indentation. That's the force we are applying to the system. So fortunately, we are dealing here with cells that have a very, very thin cortex. And the cortex is comprised of the plasma membrane, and adhered to the plasma membrane is an actin or ectomyosin cytoskeleton. The thickness is only a couple of 100 nanometers. We will see later that this is a little bit variable and depends also on the polarity. But that tells us that the stretching term compared to the bending depends on the thickness to the power of 3, meaning that if we write down the energy of stretching divided by the energy of bending, that is proportional to the radius of the cell divided by the thickness of the shell to the power of 2. And this is much larger than 1000. That means that stretching is overwhelmingly more important than bending in this case. And this means that we are left with a very simple physics here, and this is soap bubble physics, if you want. So it's a minimal surface that is kept in order to minimize the Hamiltonian here, the energy. All right, so how do we get then a force distance or force time curve? And the way we are doing that is when we have these two plates that are in parallel and compressing the cell, we have to describe the shape. And the shape is then basically described here by this prefactor, and here you see the tangent term. So that means that we have a typical pre-stress, sigma 0 here, plus an area compressibility modulus. That's some kind of a two-dimensional Hookean law, if you have to consider, times the relative area change. That's delta A divided by A0, so the geometrical area. And these are only radii that can be determined then numerically. The trick now to get from this completely elastic description to a viscoelastic description, since we have seen that this is there we have a relaxation of the force when we keep the distance constant, is that we make the area compressibility modulus time dependent. And here we use a simple power law, meaning that we have to introduce the power law exponent beta here to describe this relaxation. That's typically found in biological systems, so there's a distribution of relaxation times here, not a specific relaxation time. So if you put that all together and have a geometric factor here, g of as a function of theta, we get this kind of memory kernel here for the compression, and for relaxation we have to divide that into two parts. One is the compression and one is the relaxation part. And this then allows us to cast that into an analytic expression that we can then use to describe our data in this case, also at different velocities. That's a good test whether the theory is consistent, since the only thing we have done here is the compression at two different velocities and the relaxation accordingly. And that means that we see here with the same theory that the curves match pretty nicely the experimental data, albeit we only have used a different velocity here. All the other parameters are constant. Okay, since this is just a good start into this description of the data, we first have to think about how the cell is actually constructed. What kind of description do we really need? We have a shell, of course, that is comprised of this ectomyosin cytoskeleton, but how does the architecture of the ectomyosin cytoskeleton then be transferred to a viscoelastic response function? If you have a closer look, you can use either a STEP microscope or an electron microscope, and this is the work done together with Daniel Florman from the Lautenschläger group. He helped us to get nice SEM data, and you can see here that there's a very dense network or mesh of actin here at the surface. And you see the microvilli also pointing out, and this is a cell that just barely adhered to the surface. And the cortex thickness is around 100 to 200 nanometers. We have investigated that in more detail by extracting the cortices and then did a bubble analysis. That's a code that has been developed here in my group, but it's based on the work from Ben Fabry, who has suggested this method of placing these circles inside the mesh. And this is artificial retina of the raw data, which you see here on the right. And that gives us access to the mesh size, which is around 100, 200 nanometers, so very, very low mesh sizes here. And does it allow us to obtain an estimate for the area compressibility modulus, this two-dimensional modulus, when you stretch this shell? And for that, you need also the thickness, of course. And the thickness is then also for these cells in suspension determined by fluorescence microscopy. And as you can see, you get something like 400, and depending on the type of cells here for fibroblasts, but for MDCK II cells in suspension, we get something like maybe 200 nanometers, in confluent layers a little bit thicker. And that gives us the possibility to use network theories to estimate the area compressibility modulus. That's expectation value is around 10 to the minus 2 to 10 to the minus 3 Newton per meter. So it's very low. If you compare that with a typical lipid bilayer, you will find that the bilayer has maybe an area compressibility modulus of 0.2 Newton per meter. So it's basically inextensible, because that's a liquid crystal and is completely different as far as the architecture is concerned. However, membrane is in large excess at the surface, so meaning that this is barely stretched, but it's the network here which holds the cell together. All right, so oh, I'm sorry. The epithelial cells are, however, polar. Here we are looking at cells in suspension, but what we are interested in is how are the cells these are epithelial cells when they are fully polarized, forming an epithelial confluent monolayer. And yeah, the apical-basal polarity is very apparent if you look through a microscope, an optical microscope. You see an apical surface that is comprising microvilli. Microvilli are filled with actin bundles that basically enlarge the surface of the apical surface by a large factor. And these cells are connected to each other by cell-cell adhesion contacts, like the tight junctions here in the very top part in the apical domain. Then we have, of course, adherence junctions that are connected to the actin belt. Tight junctions are also connected to actin. Then we have desmosomes, hemidesmosomes, and here at the end, also the focal contacts. So that means the cells might be very different apically and basally. And what is the function of that, and what could be the consequences of this difference in response functions here? That was basically the motivation for our study. And the first thing we did is we compared the data we obtained from cells in suspension with those just adhered cells, single cells adhered to a surface, and cells in the context of an epithelial monolayer. And if you look now at the tension, that's the pre-stress of the ectomyosin network, you see that they're pretty much identical. The round cells are a little bit have a little larger tension. However, if you look at the area compressibility modulus, and this is a logarithmic scale here, you see that the round cells in suspension are way stiffer than single cells or confluent cells. And as a reference, we have here cells that are deformed with a round indenture and a conical one, adapting the theory. And this helps us to see that this theory we are using is indenture invariant. And what you find is also that the fluidity parameter, that is the power exponent in our theory, meaning the larger the power exponent is, it can maximally be around 1, then it's a fluid, a Newtonian fluid, and if it's 0, it's an elastic solid. You see that the fluidity is also increasing of cells that are in the context of an confluent cell monolayer. So there is a difference, but what is the difference between apical and basal surface? So before we come to that point, we have to state that the fluidity and the area compressibility are not independent parameters, but they're rather following this universal scaling law that is first found by Jeffrey Fredberg and Ben Fabry. They have investigated it in lots of detail, and has to do with soft matter properties of soft glassy rheology. I'm not going into detail here, but just want to mention that this is not an independent quantity. All right, so what can we do to interfere with the apical domain or the response of the apical domain to deformation? We can, of course, change the actin cortex directly underneath the plasma membrane. One thing is to implement additional crosslinkers. So usually the F-actin network is connected to the inner side of the plasma membrane via PIP2 lipids that are recognized by a molecule which is called azarine. Azarine has an F-erm domain, which recognizes the PIP2, and an C-erm domain that recognizes F-actin. And then it switches from a dormant to an effective crosslinker conformation and connects these two entities in a reversible manner, but very tight manner. So what we tried first is to put more azarine inside the cell by using injection, micro-injection techniques. We found that this was not possible, so we had to increase the PIP concentration in the surface using micelles. And that's what we did here. So we injected into particular cells PIP2 micelles in order, or we hope that there would accumulate at the inside of the lipid bilayer and thereby provide more anchoring points for the azarine. And indeed we could, if you see on the left-hand side, that's the typical cell that has not been injected with supplied with additional PIP2, and on the right-hand side you see the result after PIP2 injection. Some of the PIP2 were fluorescently labeled, so you can see them. And so there's an accumulation of PIP2, and if you look now with an atomic force microscope on the topography, you can see the slight differences of treated and untreated cells, which is good. That means that we haven't interfered with the health of the cells too much. But if you look now at the response and the area compressibility modulus is the most important parameter here. You see immediately here in the stiffness map that the cell that has been injected is much stiffer than the cells that are untreated. So meaning that additional cross-linkers can stiffen the network tremendously, and that actually tells us that this is a very delicate and highly regulated property. The polarity issue brought us to a point where my students, Peter Niedmann and Kevin Kaup. Kevin Kaup was an MTL student, and the first one I had actually in my group, and they had this very clever idea to look at the actin isoforms. If you talk about polarity, there are different polarity markers, but none of them are so interesting for us that they actually change the mechanical properties or would have been identified as those. So what we looked at is the isoform distribution of beta and gamma actin. These are the cytoplasmic actin isoforms. And if you look at the amino acid sequence at the N terminus, you see that the difference is negligible. So when they first arrived with this idea, they saw a paper from Moore and Balasubramanian's group in Warwick that showed me that, and this is alpha actin, this beta actin, gamma actin, and there's aspartate and glutamate here as the amino acids, meaning there's only a CH2 group difference. There's not even a charge difference here, so I would not expect a big difference here. But however, they persisted and wanted to investigate that in lots of detail, and after we have done some we have done some experiments in artificial systems. We also got the hold on a cell line that is devoid of, there's a knockout of gamma actin, and that allowed us to study that in detail, what the role of the gamma actin is in cells. And that was a collaboration with Sandra Siti from University of Geneva. And what we found is that when we're doing AFM experiments here, we see that the pre-stress is reduced quite substantially, but also the area compressibility modulus, and that is the important parameter, is reduced tremendously here when we knock out the gamma actin while the fluidity increases. That means that gamma actin depletion is leading to softer cells, so that means the stability is partly depending on the gamma actin presence. What we found is that the cortex visibly was not affected. So we did STAT microscopy on extracted cortices from gamma actin knockouts and wild type and didn't find any big difference. Small differences, but not a big difference as far as mesh size is concerned. So we looked again at our data from artificial networks. So these are actin networks made of alpha, beta, and gamma actin. And you see here the spectra we obtained from microbiology measurements, work done by Peter Niedmann and Kevin Kaup in collaboration with Moore and Balasubramanian from Warwick, who gave us these isoforms, actin isoforms. And you could see immediately here from the G prime, and these are these continuous lines here, that the alpha and gamma actin isoforms are much stiffer than the beta actin, which is predominantly found in the basal side. So we have here an important contribution of the isoforms to the mechanical properties of artificial networks as well as living cells. So this is what I called in the beginning the interface between bottom-up and top-down approaches where we work with cells, as well as artificial systems in order to prove some of our hypotheses here. All right, so that was interesting. What is the reason for that? That was, of course, the biggest question. We first were totally puzzled because there's not a big difference in amino acid sequence, but that actually matters as far as the complexation with magnesium is concerned. And we found that small changes in magnesium 2+ concentration already lead to bundling of beta-actin. So what is needed for stress fibers, of course, but not much happens to gamma-actin. In fact, it stays a network and thereby provides the stability which is needed to have a stable cortex. So that's probably the reason for this difference. That is this CH2 group that is decisive for the availability for complexation of magnesium 2+ and also calcium, of course, but magnesium is more important. Okay, that brings us now to the question, the apical membrane we can investigate very easily because that's accessible by our AFM instrumentation, but also by using optical tweezers or other instrumentations. If you culture the cells on any kind of culturing dish, you can easily measure the deformation of the apical side. However, the basal side remains totally inaccessible. And we were thinking, how can we address that and how can we measure the mechanical properties of the basal side? And if you look now at these two images, you see that the apical cortex is totally different as far as the architecture is concerned than the basal cortex. You see all these nice stress fibers here in the basal cortex that are part of the focal contacts, of course. And here you see this mesh-like structure. Please note that the scaling is a little different here. So how do we get access to that? And the idea was to change to tissue which is more native, but also polarized in a way that gives us access to the basal side of the cells that form this tissue. So if you go to, let's get the full picture here. If you go to the typical tissue where you find these cells which we're using, that are MDCK II cells, that are cells from the renal tube of the kidney. So meaning that they usually form these tubes, these hollow tubes. And this was not very difficult to get cysts from them, and cysts are then can be studied with an AFM, of course. The good thing is that these cysts form in a way that the apical side is pointing to the lumen, while the basal side is pointing to the outside. And this is work that we have done together with the group of Alf Honigmann in Dresden, and that's mostly done by Amaury Perestriado and Ulla Unkelbach from my group, and they have established the cyst formation, but also the mechanical experiments on these cysts. So this allows us now to get access to the apical side, but also, of course, to the lateral domain here. I'm not going to talk about the latter one, but I want to talk about the basal properties here. And the idea is, of course, that we show you how these cysts, you see here on the left-hand side, a cyst in solution. Here's a syringe that approaches now. And you see when we stop here for a second, no, it didn't work. Here it works. Okay, you see they're pretty resilient to deformation. And at the end, they're actually burst. Let's wait until the burst. Here. So that means that the lumen is a constant volume assumption is valid here. We have tested that, of course, but what you also see is when the syringe approaches the shell of the cyst, the cells become thinner. And that means there is also an effect that has been first described by Javier Trepar that is called super-elasticity. And we have to enlarge our theory to capture that. I show you here what I mean with super-elasticity. You see here typical cyst that has been formed, and there is a constant volume here in the cyst, so that's just a cross section. What you see here, the apical side points towards the lumen and the basal side to the outside. And you see already that the nuclei are deformed, have a very elliptical shape here. And when I release, or when we release here the Laplace pressure, which is inside this structure, then the structure collapses and forms these much thicker shells, as you can see. So the nuclei, they adopt a typical shape of a more roundish shape again. And that means that the cells, when they are deformed or when they are subject to large Laplace pressures, that they are thin. Getting thinner means that they mitigate the stress by deforming into larger assemblies. And this is important because that reduces the stress tremendously. So this kind of experiment, what we are doing, so we are with a very large colloidal probe of more than 20 microns, we deform these cysts. And what happens then is that these cysts deform, the cells of the cyst deform in a way that they are getting thinner and relaxing the tension. You see that here in the force as a function of alpha. Alpha is the relative area change. You see that this term here arises that is due to super-elasticity. It's nonlinear here. And only if alpha is very large, with changes in alpha, then you get a large impact on the stress, which then reduces due to the fact that these cells flatten. The experiment is very similar to the single cell experiment, but now we can basically deform the cells or the cysts of the cell from the basal side. That's the new information we get here. And the theory we developed with the super-elastic part, which we can neglect here for these small forces. We get this kind of soap bubble physics for larger entities here as well. And we are then able also to obtain viscoelastic properties. So you have here the pretension that's way larger than if you go for hemicysts. Hemicysts have the reversed polarity, so the apical side points to the outside. And otherwise, they're very similar, but you see that is way stiffer. So, way stiffer. And the area compressibility modulus is also higher than for hemicyst, and the fluidity is much lower. That means that the basal side seems to be much stiffer and also less fluid than the apical side. In order to get that kind of information also on a single cell level, so we used a sharp indenter and indented the cell, the cyst basally here from the outside. And we found that indeed, if we compare now the mechanical parameters from the apical side, these are in red, for the wild type and the mutant, where we knocked out the gamma actin, we see that there's a little bit of a difference here apically. So when we of course knock out the gamma actin, the tension goes down, the area compressibility apically goes very much down, and better actually increases. So that's what we have seen before. Apically, there's a very similar trend. So however, on a completely different level. You see that the tension is much larger here, meaning that the basal side is way stiffer than the apical side. And the area compressibility, please note that this logarithmic scale is also very much larger, but the trend is the same, that if you knock out the gamma actin, then we see that these cells get softer, but also more fluid. Here you see that we get something like 0.2 for beta, which is very, very close to an elastic solid. So meaning that the basal side is more like an elastic shell that covers the cyst, and the apical side, which is pointing to the lumen, is a much softer, softer surface. So what do we learn from that? So this is a typical cyst from a tube cross section from the renal tube, and what you see is that very similar to what we have seen in our artificial systems, that the apical side is a soft, squishy surface, while the basal side here is more like the elastic scaffold of the system. So that means what we see here is that an increase in Laplace pressure that often occurs if the osmotic challenges differ. That what happens is that the cells deform in a way that they're getting less elongated, they're becoming much thinner, but usually the basal side is less affected, and it gives the elastic scaffold for the whole system. So that means that these cells or the polarity enables the cells to endure very, very high Laplace pressures, but also have a shape memory that is stored in the basal side. So they are totally different properties, but also different time scales at which this happens. All right, so with that, I would like to close. Thank my group who contributed to the work, and you for your attention. Thanks.
Andreas, what I'm always curious is when you see these great presentations about the results and everything, how much time did actually went into this, of how many people? I mean, you have the last slide with all the people in the lab, but and then you have a nice story, and I know this from my PhD, right? You end up with a nice story, and then sometimes the very first experiment you show to start the story was actually the last one you conducted in the lab. How is this in this case?
That's a very good question, and it's actually showing how such a research develops. I mean, this is maybe 15 years old when we started first to look at these things, and then get the first idea of how to describe the data, how to obtain it, and yeah, it's probably 10, 15 PhDs that have contributed to the work overall. I mean, maybe one per slide or something. Not that bad, but it's quite a lot, yeah. And also collaborations, of course,
Sure.
from other universities and also our own university. That's always great, yeah.
In times like this at the moment, let's face it this way, how much you think collaborations are? I mean, have they become more important? Are they less important? Are they more difficult now in this now with when we see the situations on science and cutting down fundings and everything, would you say?
Yeah, the funding problem is hitting us hard, but I think collaborations are not yet affected by political things around us, but that will be, I mean, especially with the US, that will be a problem sooner or later. I think that's a good question. I mean, the research-wise, we are basically relying more and more on collaborations because we have very specific problems. The techniques are very difficult, so you cannot learn that, especially when you require students to finish in 3 to 4 years. I mean, this is how should that work? I mean, they're facing a very complicated interdisciplinary problem, need to learn maybe 10, 20 techniques, and this is not going to work. That's not realistic. So the collaborations are more and more important to divide the work between specialists. And I mean, theory is the other thing. I mean, you cannot do all the theory and the experiments, so you need to have theoreticians that understand experimental limitations on the other side.
Yeah, I mean, I also highly benefited during my PhD from the core facilities we had, right? There you really had the experts on one specific technique, which you can just go and they can run the experiment. You can discuss everything, and then you can see how to optimize. And this in a very much shorter time frame than if I would have needed to learn all these techniques myself and run them. But then would you say also topics like, yep, sorry.
Yeah, that's a good thing when you're working at the Max Planck Institute, where the organization is a little bit, let's say, more advanced than at the university, where On the other hand, you have the possibility to learn more techniques, maybe at the university, really with hands-on, and you have to do the experiments by yourself, can be also beneficial. On the other hand, you cannot do as much as at the Max Planck Institute then.
But then, as you also mentioned, I think it's also getting more and more interdisciplinary, right? The topics like matter to life and others, right, where you get really experts from the different fields are probably more and more important also in the future to address these different kind of complex topics from different angles, different point of views, bringing together the experts in their different discipline to then create something new.
Yeah, that's a point. Yeah. Yeah, I guess that's also very, very nice about the Matter to Life school, that also for us PIs, that we getting closer together and can do research together and share students, and students bring actually information that's not available in the group before, due to the study actually, and that makes it a good thing. Yeah.
So would you say some of your collaborations, which went now in the last years into your work, they have been established due to the Matter to Life school? Due to reputations or master thesis?
With Ulrich Schwarz, for instance, we started to work on the cyst mechanics. And since our very simple soap bubble physics is certainly limited, and so a vertex model in 3D would be really great to explain much of the things we see. So that would be a typical, let's say, thing that comes out of this school.
Well, thank you very much. There's no further questions in the chat at the moment. So I guess everybody who still has questions, of course, they can contact you and get in contact with you. Thank you very much also for this little off-topic discussion now about Matter to Life and interdisciplinary research. I think it's also an important topic, of course. But then thank you very much for your presentation and the insights in your current and ongoing research. And I wish you and also the audience a great afternoon, and hope to see you soon at some point.
Thanks for listening. Bye-bye.
Bye.